Hello, everybody. First, let me thank the organizers for inviting me to give this keynote talk. Today, I want to share with you my views on the conservation of forest genetic resources and how we can use information we already have and information we might gather in the next future to improve our ability to conserve them. In particular, focusing on what we know and might know about adaptation and resilience. The first obvious thing one wants to do with genetic diversity and genetic resources is to conserve what's unique. So, for example, a wise approach is to conserve a copy of every unique object. We currently do it already, in general, by applying, for example, anonymous or neutral DNA markers, or by collecting phenotypic values at the individual or population level. Another level of conservation and thinking about conservation is, of course, conserving levels of diversity themselves. Here I have taken a small pleasure in going back to one of the first papers in genetic diversity of forest seeds I've ever read as a student, and I have drawn a picture reporting levels of genetic diversity with isozymes in Norway spruce across Europe. And as you can see, of course, a pattern of levels of diversity emerges, and one may be interested in conserving populations or areas that store and contain higher genetic diversity levels, whatever the estimator. Another way of looking at it is maintaining high effective population size, which is a way of keeping as many, so to speak, independent genetic lineages as possible. Yet, this is maybe not sufficient, and actually we have learned also to have a different look at distribution of genetic diversity. And for example, we have learned to look at phylogeographic lineages. In this picture, we can see a representation of independent genetic lineages in a European beach from a paper from 2006 by Magri. And what we see here is that we have valuable information in the distribution of geographic lineages. And this is actually one of the most widespread ways of looking at partitions of genetic diversity. So one may want to use this information for conservation. One question one might ask is whether this phylogeographic diversity also conveys adaptive diversity. Do these lineages differ in the adaptation to ecological gradients and environmental variation? We might or might not know the answer to this particular question related to phylogeographic lineages, but anyway, this begs the question that neutral diversity is fine, but what about using adaptation as a way of deciding what to conserve and how to efficiently conserve genetic diversity? We know that forest trees show adaptation on large-scale ecological gradients. In the upper pane here, we see that there are relationships between, on the y-axis, latitude, and on the y-axis, multiple phenotypic traits for multiple species. And we can see clearly there is a correlation between region of origin and the value of the trait. Yet, while this is clearly a useful entry to partition genetic diversity and to drive conservation strategies, we have to keep in mind that adaptation is not always ideal and optimal. This is due to adaptation lag. In the lower pane, on the left, we can see that when you transfer populations from their original latitude and you move them north and south, actually populations from the center of the distribution are actually well adapted to their own place of origin, Whereas in the right part, you see that northern populations actually are better adapted to locations that are more to the center of the distribution. So they are not exactly adapted to their own environment. This is called adaptation lag, and says that actually you can't just take a local population as being ideally adapted all the time to its environment. And why is this so? Adaptation lag may have multiple origins, and among these, for example, time synchronization of a given place may be too short. This may or may not be the case considering the long life cycles in forest trees. And the second option may be the gene flow into the marginal populations is too strong from the central populations, and so they are prevented from being adapted because actually they receive genes that convey adaptation to another place. A third and interesting possibility is the fact that genetic load is too high. In the picture that is shown here, we see a conclusion from a paper from Santi Gonzalez Martinez, and that shows that marginal populations, which is gray and white bars, have higher frequencies of potentially deleterious alleles, and this is basically due to genetic drift. So this may mean that populations at the margins, which may also be the smaller one, the smallest ones with more genetic drift, may undergo high levels of genetic load, preventing selection to actually efficiently operate. So if we move from the phenotypic distribution of adaptation to genomics, we face another interesting pattern. We know from several studies that very often only a small portion of the genome shows actual signatures of adaptation on large-scale ecological gradients. Well, this makes it somehow easy to use that information to conserve genetic variants or multigenic variants that are associated to adaptation to different levels on an ecological gradient. But there may be a problem in this, that is that many adaptations may be caused or be driven by multi-locus coordinated variations in frequencies. So for example, in the picture shown here from a very recent paper, Scott Pine shows only a handful of SNPs that are showing a significant correlation with latitude out of multiple tens of thousands. And of course, we, on the other hand, expect that probably there are way more genes that respond to such a strong variation in ecological gradient. Adaptive diversity, which is resting on multi-locus responses to a selection, poses actually a problem to us. In the diagram that is shown here, I have schematically represented what may happen in the case of a phenotypic trait controlled by a single locus, where variation between population 1 and 2 is large because that's the locus that is reacting to selection. But in the case of polygenic case, the small circles with multiple colors, you may have the same difference in phenotypic traits due to very small variations in allele frequencies at each individual locus. And these changes may be too small to be detected by single locus methods. So we face a hard limit in our methods, and unfortunately, so far, there's no universal and actually efficient method to effectively detect multi-locus polygenic adaptation at the genomic level in the absence of information on the genetic control of phenotypic traits through GWAS, which is common for forest trees, but it is important to keep in mind that for the currently existing methods, that's an inescapable piece of information you need to have to run the currently existing methods. If we change scales, geographical scales, and we go to microgeographic variation. That is the cases where populations are distributed over an area where there are multiple habitats. So you have continuous populations across multiple habitats. We can observe in some cases that genomic outliers for divergence may be as numerous at a microgeographic scale that they are at larger scale. For example, in the figure here from a recent paper of ours, on the X scale, you see the microgeographic divergence, and the green color dots are the ones that show significant divergence at the microgeographic scale. On the Y scale, you see the loci that are divergent at the regional scale, and you see that there are basically the same numbers of significant dots, and there's no overlap between the dots in the two cases. So this suggests that selective mechanisms also operate and can be detected at very small geographical scales in contrasted environments. This is quite interesting and promising, and bears on the question of balancing selection actually in a continuous population, maintaining multiple genomic optima. This also shows not only in single loci, but also at the phenotypic level. Here, I am showing two cases. The upper pane is from a case around Lake Tahoe in California, where there are steep ecological gradients, and white bark pine shows divergence at phenotypic traits that is related to the ecological gradients. And besides, the authors could also show that there are genes underlying those traits that are also divergent. So coming full circle. But also in the lower pane, you can see that if you take congeneric species that occur in the same environments, and you contrast them, and you look at contrast between different habitats indicated by the different shades of gray in the plots, and you look at the same phenotypic trait in the two populations, each column representing a trait, you can see that actually there are similar patterns of phenotypic divergence. So that means that possibly adaptively based phenotypic divergence patterns somehow can be shown to be similar in different species. An interesting question is that if this is the case, and both genomic and phenotypic divergence occurs at the local scale through habitats, that means that one clever way of conserving genetic diversity, adaptive genetic diversity, is to conserve habitats themselves. So somehow conserving genetic diversity in situ requires the conservation of habitats, and conservation of habitats automatically implies conservation of adaptive diversity. So this is maybe an important information when we look at ways of using ecological information to drive the conservation of genetic resources. But we can look at adaptive variation in different ways. So far, I've been talking about extant adaptive diversity. Now we can ask a question about the potential for adaptation, so adaptive potential. And first thing we have to do is to be very clear about what we mean by adaptive potential, and we can pick multiple definitions. The one I prefer is that adaptive potential is the capacity of a population to adapt to new environments, which means that they can shift to a new environment without loss of population fitness. So in the diagram that I've drawn, you have two cases. One is the flat line, which is the blue one, for a population that has high adaptive capacity or potential, meaning that when you move it from left to right across some environmental gradient, the population fitness, which is shown in the y-axis, doesn't change much. On the contrary, the very bent curve, the red one, is the case of a population with low adaptive capacity. As soon as you move out of the optimum, you have a loss, a large loss of population fitness. This is all very sexy. The problem is that, of course, it all depends on what new environments we look at. And the second question is how to test this, how to measure this. So, of course, one interesting thing would be to say, hey, there's a new environment caused by climate change, and how well does populations react? Of course, you should test them experimentally in the new environment, but that means that you have to wait for climate change to occur, at risk of losing those populations. So we have to find ways to get out of this, and this is maybe one of the things we have to look at in the future. We can generalize this question of adaptive potential and move to the concept of resilience. Actually, what is resilience? This is a general concept that can be converted to something applicable in practical terms to actually any system. And the general definition of resilience can be summarized as the capacity of a system to not change state when facing a disturbance. So change of state can be a very general thing, but in this case, for example, it may be population avoiding collapse or something like that. The advantage of resilience is that you can incorporate processes occurring at multiple scales and on multiple biological levels, ecology, genetics, and so on and so forth. You can include phenotypic plasticity, genetic adaptation, and so on and so forth. Resilience has three components. One is resistance, that is, the system doesn't change when undergoing disturbance. The second is resilience sensu stricto, the capacity of the system of swinging back to previous state after a disturbance. And the third is recovery. This is the capacity of the system to absorb a disturbance and gradually go back to its state. It has to be, the downside of it is that it has to be precisely defined. And so each component has to be clearly declared if we want to have something useful and applicable. So that requires also some work. And while the concept is very useful and very general, we have to pay the price of developing a conceptual framework that applies to the conservation of genetic resources and a framework that works in terms of population dynamics and population genetics. So, for example, we might define resilience, and this is one possible proposal, as the resilience of forest dynamics, regeneration, self-maintenance of a population to, for example, one or multiple aspects of climate change. It is always very important to define resilience of what to what in this case. So, as I have already said, the big question is how to translate this general concept into practical recommendations and how to survey those processes in a practical way. This is something we have to develop. I would like to finish with the last section about what we can say and what we can do beyond genetic properties, strictly speaking, and what we can gather from other disciplines, other research fields, and what we can do. Actually, my idea of the description of genetic diversity is that we should not stick to genetics, and we should include other properties, like, for example, as I already mentioned, phenotypic plasticity, phenotypic variation and divergence, and ecological features of populations and the dynamic responses of populations to environmental variance. So we should try and gather information from other research fields and other types of technology. One, as shown by my previous picture, one obvious one is remote sensing. Remote sensing, satellite surveys are increasingly precise and more powerful in terms of what they can detect and the interpretation of the data we can gather and the resolution they have on the ground. So we are now nearly to the level of a single tree, basically, because we are close to resolutions of 10 meters times 10 meters on the ground. So basically, we are close to the single individual. This is something very promising because the kind of data and the amount of data that the satellite remote sensing is collecting is increasing, and it's becoming more and more useful for us. Another kind of techniques that we can get access to and may be very useful is to find ways to identify trait proxies. As I said, I consider it very important to have access to phenotypic variation and to all traits that may be useful to describe population resilience and capacity of populations and genetic resources to respond to environmental threats. But sometimes those traits are very, very important in terms of time and resources to measure. So it would be great. For example, there are multiple molecular and physical techniques we could be used. For example, metabolomics is one. We could also use GCMS, for example, gas chromatography, mass spectrography, or near infrared spectrography, which are all techniques that produce large masses of data and can be used to describe multiple aspects of a given sample. The cost of it is that it requires robust amounts of calibration. I mean, if we want those things to be meaningful to describe the traits that we are interested in, we have to find the relationship between the proxies and the traits themselves. So this is expensive at the beginning, but it pays. The payback is great. And of course, we could find ways to develop standardized genomic methods that apply across populations and across species and can be used to directly compare levels of diversity, perhaps describing diversity at particular loci of adaptive value in multiple species. So all these things open, of course, avenues to different ways of looking at genetic resources and their conservation. It is very important to keep in mind that the measure of neutral genetic diversity patterns will always be necessary because fundamental quantities like effective population size and general diversity are always will always be important to track and to study, but this is no longer sufficient in the context where we have access to methods that allow us to study also adaptive diversity. So actually we need a multifaceted approach to the conservation of genetic resources that goes beyond actual genetic diversity and that includes multiple ways of looking at population and genetic diversity and adaptation. We have big efforts to make to qualify, both from the theoretical and the empirical point of view, statements about adaptability and resilience. Those things are very valuable, but we really have to treat them with care and attention and to do the effort of defining them very, very precisely if we want them to be of any use. And of course, as I said, we need to leverage all methods and technologies that allow us to get generalized high throughput assessments of the status of forest genetic resources from different points of view. even if this comes at the expense of having a detailed characterization of individual populations and perhaps species. And what may be the link among all these things? Actually, even though I haven't gone into details about this, the slogan may be models in the sense that if we want to use, if we want to provide predictive descriptions of what is going to happen to our genetic resources, then you have to integrate all the information I've been talking about into process-based models or other kinds of statistical models that allow us to actually look forward and describe the future of our forest genetic resources. So models are a very important piece of this process, and we should invest our forces in the near future, both on our precision in collecting and interpreting data and results, and in using them for predictive purposes. With this, I will conclude, and I wish to thank you for your attention, and I'll be happy to answer any questions you may have.